Okay, good morning. Good morning. So I'm glad to start the fourth day of the course. And my name is Shai Carmi. I'm from the Hebrew University of Jerusalem in Israel. And I will talk about statistical methods for pre-implantation genetic testing. Just to introduce myself, to give some background, I mostly work on population genetics and medical genetics. Population genetics, I've been working on studying the history of populations. Also recently I got into genetic privacy. Just wanted to point out one very recent manuscript together with Yaniv Erlich from MyHeritage. You can see in this link here, where we showed that essentially half of white Americans are at risk of being identified based on their DNA alone by searching for matches for relatives in consumer genomics databases. So this is, I think, is extremely interesting, and you're welcome to check out the manuscript. What I will talk about today is diagnostics, specifically in the context of pre-implantation genetic testing. This is the roadmap for the talk. First, I will give an introduction on the problem and give some context, and then I will move on to describe the methods. I will be using hidden Markov models, and those were described already on Monday by Sharon, but I think they are quite important. So I will also give some like a quick tutorial on how to what the model is like and how to work with it. Then I will describe the methods that we developed and then show some results. Initially, I thought of discussing also some applications of HMMs for ancient DNA, but I will leave this for another time. Okay, so as we all know, like from genetics 101, if we have two parents, a couple, where both are carriers of recessive disease mutations, meaning both are heterozygote carriers, then we know that on average, 1/4 of their children will be affected. So it will be those individuals who carry two copies of the small a allele, the deleterious alleles. So this is well known. The question is what to do for a couple after an affected child was born, or maybe the carrier status was found in carrier screening, and so on. So one option is to just have another child and then just cross fingers to hoping that this child will not be affected. This is obviously not a great idea. Another option is to get tested during pregnancy and then abort the pregnancy in case the child is a homozygote carrier. What people mostly do today and over the past like 20 years or so is called pre-implantation genetic diagnosis, or PGD. So in this procedure, the couple first undergoes IVF, in vitro fertilization. In this process, oocytes are extracted from the mother, and sperm from the father, and then the eggs are fertilized on a plate in vitro, and then the embryos are being tested for this disease mutation. And only embryos that are not homozygote carriers are being implanted into the uterus of the mother to initiate pregnancy. So this is PGD. There is another type of genetic problem that causes in people doing for which people also test their embryos, and this is called copy number variants. So copy number variants, or CNVs, are loss or duplication of large genomic segments. In this talk, I mostly focus on megabase scale gains or losses. Sometimes we also talk about loss or gain of entire chromosome, and this is called aneuploidy. So, for example, here you can see gain of chromosome 21. So this person has a chromosome 21 trisomy. Do you know what disease this is? So, yeah, okay. So this is Down syndrome, right? And usually actually, what is surprising is that perhaps surprising is that for all women, even young healthy women, about 25% of the oocytes are aneuploid. As you can see in this figure here. So the rate of aneuploidy is quite high to begin with, but then it starts to grow very quickly, very rapidly with age. So for women of advanced age, or sometimes in couples where they have recurrent pregnancy loss, it might be a problem of aneuploidy. And what people do is called pre-implantation genetic screening, or PGS. That's a bit, the nomenclature is a bit confusing. And this means again, generating embryos by IVF and then testing them for chromosomal aberrations. Yeah. Okay. Okay. So how is this done, right? This is quite interesting, I think. So the embryo begins with one cell, the zygote, and then the embryo is grown on a dish for between 3 to 5 days. Different clinics differ on their practices. So by 3 days, the embryo looks like this. It has 7 or 8 cells, and then a single cell is extracted. So this is not harmful to the embryo in any known way. In fact, at this stage, we can also split the embryo by half and the 2 parts, and it will grow normally. So there is a first stage of a biopsy, taking out a single cell, and then from the single cells, the DNA is extracted, and then there is an important step of whole genome amplification, where we take the the single DNA molecules that exist in the cell, and then we multiply them, and then we have enough genetic material to do the genetic testing. So there have been many methods for to do PGD or PGS. The very traditional methods like FISH suffered from relatively high error rates. Nowadays, what most labs are doing is just testing markers in the vicinity of the disease mutation, usually STRs. The problem is there are a number of problems with this approach, and this is why it's starting to disappear. First, it requires separate tests for PGD or PGS, because this approach is not informative on copy number variants. And second, it requires the lab to develop family-specific assays. So for each family, it takes several weeks to just find which polymorphic markers they have around their mutation, and then it takes a lot of time. Sometimes they need more relatives, and it's very cumbersome. And there are new approaches using the new genomic technologies like microarray and sequencing that I will talk about soon. But currently, the best approaches, the most accurate approaches, can be expensive, and they also can take a lot of time. They can take several days to complete. And this is a problem because we wouldn't want to freeze the embryo. So we can't just let the embryo grow on the dish. We can't just stop its growth. So we would have to freeze it. If the test takes more than a single day, we have to freeze the embryo and then thaw it before implantation. Okay, so there is need for new methods. And some time ago, I was approached by David Zevi from Shai Carmi Medical Center in Jerusalem to help them develop new methods or new statistical methods for pre-implantation genetic testing. So I was very happy for this collaboration opportunity because actually I believe that the story is bigger than just helping one lab set up a testing pipeline. And the reason to understand the reasons for this, let me tell you 3 facts. So first, Israel is quite unique in having the highest fertility rate among developed countries. So the number of children per woman in Israel is 3.1 compared to 1.7 for the OECD average. So this is quite high, and Jerusalem is at the center of this fertility madness, if you wish. And Shai Carmi Medical Center, which is just one hospital in Jerusalem, had 22,000 births last year. So I don't have official statistics for that, but I looked up online, and that it would make it by a huge gap the 3rd largest in the United States. So that's pretty big. The second fact is that IVF is free in Israel. So that may sound crazy, but it's actually true, up to 2 children per couple. And the third fact is that in Israel live a number of founder populations. So half the Jewish population in Israel is Ashkenazi, and that's we and others show that that's a founder population with a narrow bottleneck from about 1,000 years ago, and that led to a few tens of disease mutations of quite high frequency. Other populations of Israel, Jewish and non-Jewish, they also suffer from a relatively high frequency of many disease mutations. So testing for carrier status is very important in Israel, and PGD is a big thing in Israel. And Shaarei Tzedek has 400 cycles a year, which makes it one of the largest centers in Europe. So the wish list for a genetic test from again from the clinicians working in the hospital is again a universal test, single test for everything we want to know about the embryo. No need for family-specific preparation, and of course accurate, fast, and affordable. So as I mentioned, there are a number of technologies that are emerging for PGT. The current most technology is becoming more and more popular is a DNA microarray. Illumina is a product called carrier mapping, and it works pretty well, but it's still somewhat expensive. And we thought that potentially next generation sequencing is going to be superior. It's going to have it could have better performance for a lower price. Usually when working with next generation sequencing, the key parameter when designing a test is the coverage. So there is an obvious trade-off between accuracy and cost. So with high coverage, we cover each nucleotide in the genome several times. So this gives us good accuracy, but of course it's more costly and it takes more time to complete the test. Low coverage, we have sometimes we don't even cover. We don't even have any read covering a certain region, but it's much cheaper. We chose to go with the low coverage because potentially it has a number of advantages. Obviously, it is cheaper, faster, and the reduction in price allows us to test the entire genome, and thereby we could test both mutations and complete number of variants, and we also don't need any family-specific preparations. So the advantage of low-coverage sequencing has been noticed. For example, there were multiple papers. There was one by Bogdan Pasenyuk, I don't see him here, from a few years ago, where he showed that the most effective way to use a certain budget for association studies is to use very low coverage, and we thought it's a good idea and try to develop a method that could employ this technology. Obviously, there are problems with low coverage sequencing. So many regions remain completely uncovered, and sometimes it's not even by chance. Sometimes there are systematic. The bias is systematic. Also, if you want to genotype sites that are heterozygous, this is a problem. Of course, we need at least 2 reads just to, on the best case, just to make sure we saw both alleles. And another problem is that's particularly severe in single-cell sequencing is the allele dropout. This means that many alleles will not even be sequenced. So they're just they're completely lost during the whole genome amplification stage. We'll not sequence them even if we had much higher coverage. So what this means is that there is never enough information to genotype each SNP individually. We'll never have confidence just looking at one SNP. So we have to aggregate information from the entire genome across entire long chromosomal region. Okay, so I'm getting close to the actual method, and just to give you kind of like the setting that we have. So usually we will have parents and an affected child. They are genotyped to high quality on the microarray. Then we phase the child using the parents. Then we extract DNA and we use coverage of 1x on average. And then we map the reads only to the array SNPs. We discard all the reads that map to other positions in the genome. And then what I will show in the following is that 2 hidden Markov models that we developed, where the first is able to identify which haplotypes were transmitted to each embryo, and thereby the carrier status of each embryo, and the second will detect copy number variants. So I need to say there are many papers, many papers with that use the similar approaches. This is pretty standard approach. What's nice about this project is the way we refined those hidden Markov models to the particular details of this noisy single cell data and achieve the clinically useful application. Yes. It's a good question. We wanted to have good quality genotypes, at least for the parents. Maybe you can eliminate one child, but at least for for the parents, if we have only low coverage sequencing for the parents, it would have made it very difficult to obtain high accuracy for the embryos. No, the microarray. No, we are not. Sequencing the parents again. We are doing microarray on the parents and one child, and low coverage sequencing on the embryos. Okay. Okay. So in the next part of the talk, I will describe hidden Markov models. So again, you have heard about them probably in multiple talks, but I thought that would be a good opportunity to give a quick tutorial about what they are and how do we infer in hidden Markov models. How many people know about hidden Markov models well? Almost everyone. Okay. Okay, maybe I'll make this part quick. Okay, just like a quick reminder. So hidden Markov models are mostly used for analyzing sequential data. And the idea is the following. We have a system, and the system is characterized at each time point by an internal state. So the state here is denoted by xt. And the idea is that we don't know the states. The states are hidden or latent, but we have data. So the data or observations are denoted as yt here. And we don't, and we see, and each observation at each time point depends on the state. So while we don't have information on the hidden states, we can learn about the identity of the states from the observations. The states themselves, they evolve as a Markov chain. So xt depends only on xt minus 1 on the state at the previous time point, but not on the state at any other time point. To give like a simple demonstration of where this is useful, so we heard one example by Sharon on Monday, where the states were the pairing status of the RNA. Here I describe a simple example. Let's say a person has an internal state. A person can be happy or unhappy. And whenever a person is happy on one day, it has probability 0.95 to remain happy for the next day, and probability 0.05 to become unhappy the next day. And similarly for the other state. And so these are called the transition probabilities. But let's say that we don't observe these, we cannot observe this internal state. Let's say we are bartenders, and we see which drink was ordered each day when that person went to the bar. So let's say there are 3 possible drinks, and the key point is that the probability to order each drink depends on the internal state. So let's say if I'm happy, I have higher probability to order wine and low probability to order vodka, and vice versa if I'm unhappy. So let's say I work at the bar. And I would like to tell something about the internal state of this person that I see every day, but I don't know much about except like a kind of observation of what was ordered. So these are called the emission probabilities. And again, the key point is the emission probabilities depend on the state. They're different for each state. So what can we do with hidden Markov models? So given that we see a sequence of observations, for example. Like in Sharon's example, we saw a sequence of signals from the shape-seek method, or we see the sequence of drinks that were ordered. We can infer the sequence of states that has the highest probability. This is accomplished by the Viterbi algorithm, which I will actually explain in detail in the next slide. We can also compute the probability to be in a given state at a given time point. This is called the forward-backward algorithm. And we don't even have to know the parameters of the model, the transition and emission probabilities. We can also infer the most likely parameters using the Baum-Welch expectation maximization algorithm. So I thought, for the purpose of the tutorial, to explain just the Viterbi algorithm. So let me give some definitions. So let's say we have k possible states, d possible observations, and total time t. So the transition probabilities are denoted by aij. So this is the probability that the system goes from state I at one time step to state j, the next time step. The emission probabilities are denoted by bi of y, and this is the probability to observe y, given that we are in state I. And we also have initial state probabilities for the first time point. So the Viterbi algorithm works like this. So the problem is the naive way to find the most likely sequence would be just to consider all possible sequences. The problem is the number of sequences, number of possible sequences increases exponentially with the total length, with the total time points, total number of time points, and that's not very useful. But the point is that we don't have to examine all sequences. We can just we can achieve linear time, and we do that by keeping track at each time point the best path that led to each state in that time point. So, for example, suppose we have 4 possible states, 1, 2, 3, and 4, and we have time t and time t minus 1. So the best path, the path with the highest probability, first we compute what is the probability that the best path go through state number 1. And if you think about it, for a path or for a sequence to go through a certain state at time t, it must go through one of the states at time t minus 1. So we just have to find the state at time t minus 1 that gives the maximum, the highest probability for the best path to go through state 1. And then we do that for each of the other states of the model. So mathematically, if P of j is the probability of the best path to end at state j at time t, we can write a recursive equation for Pj of t. We can calculate it using the probability of the best path to pass through each of the possible states in the previous time point. So we just have to maximize over all the states in the previous time points, then we multiply by the transition and emission probabilities. This way we can compute this probability for each time point until we reach the final time point. And then once we are at the end, we just take the state with the highest probability, and then we can trace our way back to the beginning of the sequence along those pink arrows. Every time we just go backwards to the state that gave us the highest probability. So this gives us the Viterbi sequence. And the running time of this procedure is linear in the number of data points, number of time points, and squared in the number of states. Okay, so with that introduction, now we can move on to describe the results for the methods for PGT. So just to make sure we are all on the same page, I want to mention what is a haplotype. So what I call a haplotype is a sequence of alleles all coming from the same chromosome, either over a short region or entire chromosome. So the sequence of an individual can be represented as 2 haplotypes, the maternal haplotype and paternal haplotype. And the idea is, for the first HMM that I will describe, is that the hidden Markov model, the hidden states of the HMM are as follows. So for each SNP, we have for each embryo, we have the state is 2 bits. One bit describes whether the maternal haplotype of the embryo is the same as the affected child, and the second bit is the same for the paternal haplotype, the one transmitted from the father. Okay, so if these are the parents, as we know, the chromosomes of the children will be a mixture of the 2 chromosomes of each parent, and we simply track which chromosome was transmitted. So how is this helping us? Is because suppose we have the given state, suppose we have estimated the hidden states, this way we can tell if immediately if the embryo carries the same genotype as the affected child or not. And this is a useful way for us to determine whether an embryo is good for implantation or not. Now, in this model, the transitions are simply recombination events, okay, in the parents transmitted to each embryo, and the probabilities of transitions are determined by the genetic distances between SNPs. This means we measure them simply according to the genetic maps in centimorgans. The distance between 2 SNPs in centimorgans directly corresponds to the probability of a crossover in the previous generation. So this is straightforward. What's more complicated is the emission probabilities. So first of all, what are the observations in the model? So what we observe is the read count. So so how does it work? So let's say this is one SNP and we aligned all those reads to the genome with that particular SNP, and we observe 2 reads with A and 1 with G. So our read count is 2 and 1. So that's the observation that we have at each SNP. And now we need to model the emission probabilities. So once we have the state, once we have the transmission state, we automatically immediately we have the genotype for each embryo in each SNP, And the question is how to model the read counts given the genotype. And there are several issues. First, we'll have to do it separately for homozygous and heterozygous genotypes. And second, we have to model errors in either platform. So let's start with the heterozygous genotype. So let's say the true genotype, meaning like the state at a certain position is heterozygous. Let's say it's AG, and let's say we observed 5 reads. So a naive model would be for the read counts would be binomial. So if we have 5 reads, the number of A's we expect it to be a binomial random variable with parameters 5 and 0.5, because each allele is equally possible. The problem is, because this is single-cell sequencing data, we have very strong overdispersion. So let me demonstrate what we see. So this is so here I'm showing you five plots, one for each depth or coverage. Meaning here we have only one read covering the site. Here we have 2 reads covering the site, etc. And this is showing the distribution of the number of reference alleles that we observe if the distribution was binomial. So for example, if we have just one read, it can have probability 50-50 to be either the reference or the non-reference allele. Actually, it's not exactly 50-50, but we observed a slight reference allele bias, where the probability of observing a reference allele is a bit higher. But as you can see, as the coverage increases, we expect this kind of normal, we expect the distribution to approach this kind of normal bell curve shape. What we see in practice is very different. We see the distribution as this kind of U shape, where even for higher coverage, we almost never see both alleles. Usually we see just one of them. So to model this, we use the beta binomial distribution. So the point of this, the idea behind this distribution is like binomial, but the probability of success is allowed to vary. Is by itself a beta variable, and this gives good fit for the data. To estimate the parameters of this distribution, yes? Yes. So I mean to generate this figure. So we focused on sites that we know from microarray data or high coverage data that are heterozygous, exactly. The beta binomial is just I mean that's how it looks like. I mean it's more dispersed. It has higher variance. What? So people use these kind of distributions. Usually not. There's no. They don't derive this distribution. They're just good fit for the data. But I mean, it should be. I mean, mostly when there is a process that should be binomial, but it over dispersed, this is something that people use. Yes? Okay. It's possibly because of again dropout, because when you start with a single cell, you have very little genetic material to begin with, either DNA or RNA. So if you, I mean, if it was lost during amplification, then you will not see it. Okay. Okay. So we learned the parameters of the beta binomial distribution from the data from sites that are indeed were confirmed to be heterozygous in a different experiment. And we also need to model errors for homozygous genotypes. So for example, if both parents are homozygous for A, and we see G either in the affected child or in one of the embryo, this indicates an error either in the parents or in one of the children. So for this purpose, so the probability, so if the true genotype is AA, then we would not observe only A's, but we also have a small probability to see an error. And we also, I mean, this also, it's a bit more complicated because it also allows the genotype to change if we have errors in the parents. But anyway, this I think this defines the hidden Markov model. And once we have defined the transition probabilities and the emission probabilities, we can use the Viterbi algorithm that I just described that given the data, given the read counts at each SNP, we can infer the most likely hidden states. So this is the hidden Markov model that we used for determining the haplotypes, and thereby the carrier status for the given disease mutation. Now for the copy numbers, the HMM is quite different. The idea here that the hidden states are the number of DNA copies in each genomic window. Yes? Because it's the hidden Markov models are useful whenever we have a process that changes along the genome, changes kind of slowly, and we can model the transitions between the different states. And this is very natural for this kind of problem because we can model the changes between states. They are precisely recombinations. And once we have for each state we have a different genotype that we expect, and then we can model the read counts according to the genotypes. So I mean, possibly there are other ways to do it, but I think it's a very natural model. It was also used by others in this context. Okay, so what would be the state? So the states are the copy numbers. So the normal copy number is 2. This is where an individual is diploid. But then the copy number can be 3 or 1 if we have a copy number variation or aneuploidy. The observations are the normalized read counts for each genomic window. So because this is low coverage, we cannot go for we cannot identify very short copy number variants. We have to be in the genome in windows of 1 megabase, and so these are the observations. Simply the read counts are simply the number of reads that were mapped to each genomic window. So there is this is not so. The problem is this is not so simple, and we had to do some normalization. So first of all, the coverage per each window varies quite significantly with the GC content of the window. So we had to correct for that, and even then there remains a variation, natural variation along the genome in the coverage. And to correct for that, we used for each embryo we used a number of reference samples that are known not to have any copy number variants or be euploid, and using them we normalized the read counts. So after so now we have the normalized read counts as our observations, and now we need to model the transition and emission probabilities. So for the transitions, we just came up with kind of like reasonable parameters for the probabilities of having a copy number change and the length of the copy number variant. For the emissions, we had to model the number of reads, the coverage of each window, given the copy number state. So again, we have overdispersion. So this is the empirical distribution of the number of reads, the normalized number of reads, for individuals where there is no copy number variation. So since this is a count, just the number of reads, the naive model is Poisson, but as you can see, there is very big overdispersion here, so Poisson is not a good model. We use negative binomial model, which is again kind of like empirical model that people use for overdispersed Poisson. We could have used normal or actually other distributions. One key point is that once we have this distribution of the number of reads given the copy number, to properly generate the distribution of the number of reads given the copy number state, we need to adjust to shift the distribution. We have to shift it left for a state of loss, where we expect to have half the coverage, and to shift it to the right, where we have a gain, and we expect 1.5 the coverage. And we also had to adjust the standard deviation. It turns out the standard deviation depends on the mean in a complex way. We had to adjust for that. But eventually, that defined the transition and emission probabilities and the hidden Markov model. And again, we used a Viterbi algorithm to obtain the most likely sequence of copy number states. We also used the forward-backward algorithm to compute the posterior probability of being at each state at each time point. For the last section, I will show some results. So this is what the results look like when you try to visualize them. So so we have, so let me walk you through this. So we have 22 chromosomes plus the X chromosome, which we also implemented. And for each, so for each chromosome, what I'm showing here is results for 3 children. And for each child, we have a pair of bars. So what do we see here? So the left bar corresponds to the paternal chromosome. So it will have dark shade if the paternal chromosome of the embryo of the child is the same as the affected one, and light colors if it's different. And similarly, for the right bar in each pair, which corresponds to the maternal chromosome. So this is just a quick way to visualize the results. What they do in the PGD clinic is they developed a browser for the lab technicians to use that looks like this, kind of like user-friendly browser. And this is showing a number of pieces of information. So so here at the top, what we see is the probability that the embryo has the same chromosome as the affected child. So this is the posterior probability computed using the forward-backward algorithm. What we see here, this bar here for the mother and this bar for the father, shows the Viterbi sequence, the most likely sequence. So for example, what we see here is a recombination event. So the embryo had the same chromosome as the affected child, and then there was a recombination event. So this is just one nice example. This is the BRCA2 gene, which is a risk factor for breast cancer. And we can see that even though there was recombination event very close to the gene, we are still able to very confidently assign the genotype to the embryo here. So to do more proper validation, we didn't use embryos. Now we're in the process of getting the ethical approval for that. We used actually like families, living families, and we had 3 families with 10 children overall. From each individual, from each of the children, we extracted 1 cell for the single cell sequencing. And then to validate the results, we compare them to high quality genotype that we obtained from bulk DNA, not from a single cell. So the way we look at the results is the following. We using the posterior probability, we only consider sites that are high confidence, and so in those sites the accuracy is nearly perfect. The key question is what is the proportion of the genome that is high confidence, that we can confidently determine the haplotypes. So we found that across the families it ranged between 90 to nearly 100%. Initially it was a bit frustrating. We thought, okay, why do we have relatively low confidence in 10% of the genome? Then after a while we found that the blind spots of the method are regions where either the parents are homozygous, they have long run of homozygosity, or what's even more strange is that both parents are genetically completely identical, but they're heterozygous and identical. So they have so-called IBD2 in that region. So in these regions, we have no way to tell which haplotype came from which parent. But on the other hand, the genotypes are easy to determine. So what we're doing now, writing a patch that will allow us to genotype the embryos properly, even in these regions. For the couple number variation, this is what the results look like visually. So, for example, in this one family, we found a duplication in part of chromosome 3 and deletion of most of chromosome 21. And for validation, we used an established technology of Illumina called VeriSeq, and we found 100% concordance for these copy number variants. So what we saw is that using hidden Markov models, we were able to integrate information across the genome and to provide accurate results even for the noisy single cell data. What we saw in simulations and experiments that we expect to reach to still obtain accurate results, even if we lower the coverage up to around 0.5x. Theoretically, we think we may even be able to do better at some point, but with the current algorithm, that's the performance we expect. Now, I started with the lab clinicians. They wanted to have an affordable test, so So we did also like the economic calculations of how much it's going to cost. So we found it's going to be around $2,000 per family for 8 embryos. These are the prices of a lab in Israel. It may not be representative of prices elsewhere, but that's at least what we can get, the best we can get. And this represents a 20% saving over current state of the art. But we believe this can be improved. For example, we may not have to genotype the parents if we can phase perhaps the embryos with reference data, which we have quite a lot, at least for the Ashkenazi population. And we can also combine HMMs of the CNVs and the haplotypes. This is expected to give us much better resolution for better resolution and better accuracy for those for detecting both. Before I conclude, I just want to show you. I want to show you 2 slides about one variant of the hidden Markov models that in a different project we recently worked on. So what I showed so far is a case where we have. The way I presented the hidden Markov model is that we have one chain. We have one state that evolves with the time. But very often we have situations where the hidden state is actually comprised of multiple states. So what we have is actually like M chains evolving in parallel. They are completely independent of one another. So the state at chain 1 depends only on the state at the same chain at the previous time point, and same for the other ones. On the other end, we can have one or more observations, but the observations depend on all the states. We cannot factor this, and this makes it complicated. I mean, the naive model would be quite, the running time would be too high for useful applications. Even though this kind of model, the Factorial HMM model, is expected to be very useful in genetics because it can track multiple processes. So for example, it can track the ancestry at different individuals or different generations. So for example, the first use I know in genetics is the classic Landry-Green algorithm. In our case, we expect this kind of model to track recombination in different, both recombinations and CNVs. The problem, as I mentioned, is the running time of Viterbi and the forward-backward algorithm is square in the number of states. And the number of states by itself is exponential in the number of chains. There were algorithms. There is a classic algorithm that was developed about 20 years ago to reduce the running time to nearly linear, to n log n, basically. And recently with Regev Schweiger from MyHeritage, from the industry, we developed a Python package that's user-friendly and modular that implements all the standard HMM operations with exact solutions and linear time, and you can find this package here. So I concluded. With that, I conclude and thank my collaborators on these projects and also advertise positions. Yes. Mm-hmm. Mm-hmm. So we could, copy number variants appear in all sizes. So, so we are unable to detect with low coverage. We are unable to detect the shorter ones of 100 kb, even 1 megabase would be challenging. But have in mind that they are not so important for this purpose because people actually in the PGD community debate a lot about it. Like even if you find a deletion of 5 megabase, there are a lot of healthy babies that are born with 5 megabase deletion. Actually, as much as I remember, most of them are healthy. So maybe we want to use this embryo. But we have to use. That's kind of like the minimal window size that we could use. Mm-hmm. The reason why we use this model is again because there is a correlation between regions of the genome. So if we have so again usually we're looking at long CNVs. We expect that if one window has let's say a deletion, the next window will have quite high probability of remaining in the state of deletion. Sorry. We have too little data to do it now at this point. If this starts working routinely, we'll have like 400 families a year. So maybe in 2 years I can answer that question. Yes. Yeah. Uh-huh. Yes. Right. Yeah. So as I said in the beginning of the talk that these models are standard. We also we indeed built on this model. You're talking about Roach et al. Paper. So we built on that model. The tricky part was to model the emissions, again, because of the noisy single cell data. We didn't look at it yet. Again, we will be able to better answer these questions in a couple of years when we have hundreds of data from hundreds of families. Okay.